Hello and welcome to this video. In this video, we will be discussing the topic of DNA replication, and we will be discussing what it is, what is the purpose of this process, when it happens, why it happens, and the different players that have intervention or some sort of interaction with the DNA molecule in order to achieve making this particular process. You may recall the cell has a particular lifespan, let's say, or different stages of life, and how this cell is actually growing, developing, reaching certain level of maturity, and eventually getting to divide and to have uh, this cell division process that we've seen previously, known as mitosis and as meiosis, or meiosis, depending on which type of cell is going to be the process that it actually goes through. But in order to be able to actually divide itself and to have all this genetic information still within the daughter cells when it eventually divides, we need to guarantee something that is of the utmost importance, which is going to be the genetic material, right? So when the cell is starting to grow and to reach its level of maturity, it goes through two different growth phases that we've seen previously that are gap phase 1, gap phase 2, or G1 and G2. And in this process where we have the gap or the growth phases, in between, we have something that we call the S phase or the synthesis phase. Now, before we have mentioned this, and probably you memorize it as the synthesis phase, and this is where DNA replication or DNA synthesis occurs. So this chapter is basically to look under under detail what is actually going on in this S phase and how is it possible that in gap phase 1 or G1, where we have a single-stranded chromosome, eventually reaches to gap 2, where we will have the double-stranded chromosome. Or in other words, we have a exact copy of this single-stranded chromosome that we had in G1 stage. So to further go into detail, when we talk about DNA replication, we're talking about how DNA itself will reach a duplication or a copy of all the genetic information before actually reaching cell division. In other words, we need to prepare all the genetic component so eventually and later down the path, when we do mitosis and we split the chromosomes in anaphase and we go into telophase and divide two different nuclei with all this genetic information, one for each daughter cell, well, we need to actually have available all of this genetic material so it's enough for both of these cells to have exactly the same genetic material, so each individual cell will be able to function properly and independently one from the other one. So before getting into that, which you may remember, we call it the M phase, right? Which is M phase G1. So before getting to the M phase, we need to go through the DNA replication stage, which is the S phase or the synthesis phase, right? So we can continue to grow and develop and prepare to reach the stage of cell division. So prior to that, before actually getting to that point, we need to do the duplication of the chromosomes or the duplication of all the genetic resources that we have. And this is exactly what happens in S phase. This is something that we call DNA replication, right? So overall, the context of this is that we are preparing the cell to have enough resources, so eventually during cell division, being mitosis or meiosis, then we can actually achieve it and the cell to be complete and full with all the genetic information. So very similar to what happens in mitosis, where we start with one cell and this will create two cells down the line as we divide. In here, we're going to have something very similar happening, where we will have one parent or paternal DNA strand, or one molecule that acts as the before, and with this information of the parent strand DNA, we are going to actually use this double-stranded strategy or configuration in order for us to be able to make a new strand based on the information that is available with this original or old, let's call them old strands. So as you can see, in here, we take one of these blue strands as a template. This word is important. This template, meaning like an original guide or an original set point or original set of information where we can actually start to take as a reference to make the new one. Because if we consider that DNA has two strands and they are complementary, remember that A binds with T and G binds with C, T being exclusive for DNA, then we can assume that if it this old strand we have G, then the new strand will have a C, right? And if you look at the other complementary strand that comes over here, we have a C over here. So that means that the complementary would be G, which kind of makes sense if you compare this one with this one. So by making these new strands, taking as information, or the original information, the old strands, we can have a new daughter DNA being made from an original old strand. And this characteristic, or this way of working, where we have an old strand being as a template for the new strand, this is something that is called, that is a semi-conservative process, or a semi-conservative procedure or formation of keeping the information partially the old information in there, but using it to create a new daughter cell, so we can have both daughter cells being daughter DNA molecules being created in here. So from one parent DNA molecule, we will eventually have two daughter DNAs or daughter molecules being formed as a result of this semi-conservative process. Now, looking at this more specifically, in the sense like who or what are going to be the specific players to play a significant role in here, we need to understand two things. One, that before actually getting into place or getting to perform all of this process, because DNA is in a double helix and is double stranded and all that, the first thing that they must happen is that this double stranded or double helix must be able to separate. We're gonna separate them, or like split them in half like a zipper, and through this mechanism we will be able to use this information to make the complementary bases and eventually reach to the double-stranded configuration. So who's going to do what in here? Now, we can see this diagram, which is including a lot of information, and we can organize all of this into different steps, which is an oversimplification of what's actually happening. But we can summarize all of this into 4 steps. So the first step is to separate the 2 strands. And this is going to happen with an enzyme that is called DNA helicase. You can say that this helicase enzyme would be step number 1. By the way, what's an enzyme? So an enzyme, let me put it in here. An enzyme is a protein, a type of protein that will be in charge or responsible to make the chemical reactions faster. This is an oversimplification of what an enzyme is, but it's a protein that will make chemical reactions to occur faster. So what do we mean by enzymes? Is just a thing, a molecule, specifically a protein, that will have a very specific purpose. And this first step is called the helicase enzyme. And the very specific purpose of helicase is to unzip the double-stranded DNA molecule. So thanks to helicase, we will have one strand being over here and the other strand being over here. So just like a zipper, we can say we will split it into two strands. So that was step number one. Now, step number two is not shown in this diagram, but let me put it in here. See this red square that I'm placing in here. Let's say that this thing is called a primer. And this primer will be placed by an enzyme that is called a primase. You may recall from previous videos where we have said that the SEE is code, this suffix that we see here, and in here is a suffix for an enzyme. So primase basically means that it's the enzyme that will be responsible to create the primer, right? So this enzyme would be step number two. So what is the purpose of this enzyme called primase? So even more than actually creating the primer, this primer or thing that was created by the primase is the mark or the spot where the whole DNA replication will start or will begin to happen in this particular strand. So it's kind of like a start signal where the next step, step 3, which is this enzyme number 3, is called DNA polymerase. So this DNA polymerase is an enzyme that is in charge of making a polymer that is called DNA, right? So when DNA polymerase finds the primer set up into this strand of DNA, this is going to be the signal that provides the start trigger for DNA polymerase to begin working. And what is this enzyme going to be making? It's going to move like a train over a railroad, and will start to set up complementary nucleotides. Each of these units that you see here is a nucleotide. So it will start to paste with the complementary nucleotides on the already existing DNA strand. So you can see that in here, DNA polymerase placed T-thymine with A, adenine with thymine, cytosine with guanine, and all that. So as this enzyme moves forward, just like a train, it will continue to set up nucleotides in each of the complementary nucleotides that is part of the old strand. And with this, we have 3 steps. The final fourth and final step of all of this, it's also not shown here just as primase, but the last step is something that is called the ligase. So this enzyme, DNA ligase, the name kind of suggests that it's going to be linking, or it's an enzyme that will link what? It's basically, to oversimplify this, is to link these nucleotides that were already set up, right? So while DNA polymerase makes a link between the nitrogen bases, like here, joins forces here with AT, CG, AT, and all that, ligase will finally make this overall link between the nitrogen bases to solidify all of this as one simple or single chain. And with this, we'll basically end up with one daughter DNA molecule over here and another daughter molecule over here. So overall, this process begins with a parental DNA molecule that will go through steps number 1, 2, 3, 4 to achieve having the complete replication process being complete. And now we have the two new strands, right? So just like mitosis, we have one and create two molecules. So now let me just show you an animation which is, made by DNA Learning Center. Let me just move myself here from here to here, which this source of animation is really good. I highly recommend. Shout out for DNA Learning Center. And here, if you click in the resources, you would have here biology animations. And over here, you can see the mechanism of replication. There's other cool stuff in here like transcription, translation, and many other things that we have seen in this course and some others that we still haven't seen. But if you click in here, mechanism of transcription, this is a very cool animation that it shows pretty much what's going on in this whole process. Now, this is more complex than I was just explaining, but the main concept remains where we have one, let's see here, we have one enzyme that splits the original strand into two strands. One will be continuously being replicated and continuously being applied the nucleotides to make the new strands of DNA. And the other one will go through a more complex process that we will see maybe in another course if you continue to go through higher level of biology. But the point is that from this splitting and by complementing the nucleotides with the old strands, we have two completely new sets of DNA in here. So at the end, all of this basically is an effort, and let me just go ahead and connect this with previous knowledge that you may have or you may remember into going from the single-stranded chromosome that we may find, for example, like in G1 stage of the cell cycle. Then it goes through DNA replication in the S phase to reach the G2 stage where we have the double-stranded chromosome. And then the next part of the cell cycle would go into division, right? Cell division, which means the M phase. And this is basically how during anaphase, we have the splitting of the sister chromatids, remember that? And we split them to opposite sides of the cell. And each daughter cell will contain one strand or one sister chromatid, and we begin the cycle again with each daughter cell starting again with G1. So this is just a small portion of the whole cell cycle that we call DNA replication, which is basically going from the single-stranded chromosome to the double-stranded chromosome through the 4 steps that we previously discussed. So that's pretty much the gist of this DNA replication aspect. So what are the main takeaways from this? Again, is the overall understanding of what this is for, when this is happening according to the cell cycle, what is the purpose of this, and what are the main players that are taking part of this process of DNA replication. So hopefully this was of use to review these concepts or to study some of the most important concepts if in an upcoming quiz. And let me know if you have any questions or if I can help you with something else. Thank you for watching, and I'll see you around.